Hello, and welcome back to the Mechanochemistry discussions. This seminar series is hosted by the NSF-funded Center for Mechanical Control of Chemistry, or the CMCC. I'm Ashley Martini. As always, the goal of the seminar series is to bring the community together. Our seminars stream live on the third Thursday of every month at 10 a.m. Central on the YouTube channel. The YouTube channel is also home to all of the previous presentations as part of the Mechanochemistry discussions. We'd also like to let you know about a few upcoming presentations that we hope that you will also join us for. Before we get started, a great big thanks to Dr. James Batiste, the Center Director, Jennifer Belsick, the Center's Administrative Coordinator, and two CMCC students, Quintarius Moore and Kathleen Floyd, both of whom helped coordinate the seminar series. Thank you ahead of time for joining us. Please do subscribe and follow us on YouTube and on Twitter. Lastly, quick note, this seminar is being recorded. If you have questions during the seminar, please email them to us at cmccdiscussions@gmail.com, or you can post them in YouTube. Either way, they'll be propagated to the speaker to be answered at the end of the presentation. Finally, it's my pleasure to welcome today's presentation, given by Dr. Karthik Nagapudi. Dr. Nagapudi got his PhD in polymer science from Georgia Tech in 1999 under the supervision of Dr. Haskell Beckham. He did his postdoctoral work in the lab of Dr. Elliot Chaikoff at Emory University. After his postdoc, he joined Merck Research Labs in the Physical Measurements Group in 2003. In 2011, he became the head of the Solid State Group in AgMen. In 2014, he moved to Genentech as a senior scientist in the small molecule pharmaceuticals in the Pharmaceutics group. He was promoted to senior principal scientist in 2018, and he's currently leading the biopharmaceutics, drug delivery, and a portion of the formulation development group at Genentech. His primary research interests are in the areas of mechanochemistry, application of solid-state NMR methods in pharmaceutical analysis, drug delivery technologies, and understanding the thermodynamics of phase behavior in pharmaceuticals. He has published 80 papers and has 8 patents to his name. And today it is our pleasure to have him part of the mechanochemistry discussions.
Okay. Hi, everybody. I'm Karthik Nagapudi, and today I'm going to be talking a little bit about high-throughput screening and scale-up of co-crystals using resonant acoustic mixing. Before I start, I want to thank Ashley and the organizers for giving me this opportunity to present at this forum. And I should say I'm super excited about this NSF Center for Mechanochemistry, because this is something that I've been working on for the past 10 years. And it's very interesting to see how the community has evolved and come together with this new center. So it's really a great opportunity to speak at this forum. So I'm going to talk today about co-crystals, which are pharmaceutically relevant materials, and how mechanochemistry in the form of resonant acoustic mixing can actually help to make these materials on both small and large scales. So before going into the details of the mechanochemistry work, I want to set the stage for what are the issues that we face during drug development. And I belong to a world where we do synthetic drugs. That is, we make the drugs that you typically get in your CVS or Walgreens, like acetaminophen and things like that. So we synthesize these drugs using chemical approaches, and then convert these drugs into dosage forms like tablets and capsules and so forth. That then is sent into clinical trials and finally into commercialized drugs. One of the problems that we are facing in drug discovery increasingly in these days is the use of high throughput methods have been used in drug discovery, which are really great because they try to produce a lot of candidates. But what happens is that we really optimize the drug candidates so that they can get into the cell, right? And the components of cell membrane are lipophilic. So which means that if you start to optimize for these properties, your drug molecule also becomes highly lipophilic, and they have really low water solubility, right? And that's a big problem, because when you actually take your tablet or capsule, whatever dissolves, in your gut and is the one that is transported into your systemic circulation. That is, only the dissolved component of the drug will move into your systemic circulation and then impact the disease that you're trying to impact. So if you have drugs with really poor solubility, it's barely enough to get the drug on board to do what it's supposed to be doing, right? That's a very big problem. So what we really have now is compounds with great receptor affinity and selectivity, but with very poor solubility. And these are not considered ineligible for development because they think that people in development like us can come and fix the problem of solubility, right? So the conservative estimate is these poorly soluble compounds now make up nearly 40% of the entire portfolio of new molecules in major pharmaceutical companies. So you can see it's not like some small problem, more than half the drugs that come in that we have to develop have very low solubility. So then the question is, how can we start to improve the solubility of such molecules, right? And of course, like I mentioned, these poorly soluble molecules hinder drug development in both the preclinical space when we are testing the drugs on animals or for looking at toxicology, as well as in the clinical space when we take it and test it on humans. Because if there's not enough drug on board, you cannot do any good pharmacology or any good toxicology because the drug is simply not there for you to test it. So so the whole effort in the last decade, 2 decades in the pharmaceutical development field has been to really tackle these poorly soluble molecules. How can we improve the solubility of these molecules? Right, that's been the key question. So when we do that, when we look back and, so one equation that I'm going to put up here, this is an old equation that is developed by Noyce and Whitney and somewhere in the early 1900s, where they looked at the dissolution rate of the drug. What is the dissolution rate of the drug? dQ by dt simply is the dissolution rate of the drug. And what parameters does it depend on? It's simply a fixed law of diffusion for those who are familiar with it, right? So what it really depends on under sink condition is that, when the drug has, starts to diffuse across a drug boundary layer and then goes into solution, right? This is where your drug is. You have a diffusion coefficient, and that's what this D is. And then you have a surface area of the drug, which is governed by its particle size, right? Smaller the particles, greater the surface area, and the actual solubility of the drug substance, right? What is the equilibrium solubility? So we really cannot change the diffusivity of the drug substance because in the GI tract, when we take it, whatever the diffusivity of the drug is, it is what it is. And the boundary layer thickness, there's not a whole lot of we can do. But what we can do is we can start to play around with the surface area of the drug and the solubility of the drug in order to improve the dissolution of the drug. And so therefore, what we have developed is a slew of technologies in the last decade or so to affect, how we can change the surface area of the drug. We can change the particle size of the drug. We can make it nanoparticles instead of big particles. Or we can change the solubility of the drug by changing the form of the drug, which I'll get into my next slide. So I just thought that, as we're proceeding into this, it's an interesting story. I put these two gentlemen here, Noyce and Whitney, just to give you some historical perspective. For those of you all who are very very familiar with chemistry, Noyce did his PhD with Oswald of Oswald ripening fame, right? And then he became eventually president of MIT, and he went on to be in the National Science Foundation Academy and so forth. And Whitney is a very interesting person because he's considered the father of industrial research. He really started the GE research labs, right? So these are people who laid the foundation for a lot of things that we see today. May appear very simple kind of equations, but these are kind of people who set the stage for, and even today we use these work quite extensively in industrial research. So just wanted to start with a little story before we get into the actuals, right? So in the next slide, I'm just going to show you a little bit about the different kind of methods that are available for improving solubilization of the drugs. I'm really not going to get into the various methods, except to tell you all that, there is a wide variety of things that we can do, which are the tools that are in our disposal, at our disposal to improve the solubility of these drug molecules. For example, you can think about it, most of these small molecules are crystalline in nature. You can think of making a salt of the material. You can take, bring in HCl or, some kind of counter so that it improves the pH solubility of the material, right? Or you can put it into a surfactant and, like a soap and, solubilize the material and then, administer it to the patients. There are a lot of different ways. You can put a co-solvent like ethylene glycol or something like that, or a cyclodextrin complexation. But today, what I'm going to talk about is one such method, and this is called co-crystallization, where you take this drug molecule and bring in another co-former, which is highly soluble typically, and put them together so as to give the drug enough solubility and other better properties, right? This is not a salt. In salt, you have ionic interactions, right, where you bring in the second component and it's charged, and there is proton transfer from one component to another. This is simply what we call as materials that are stabilized by hydrogen bonding, or co-crystals. So I'm going to focus on co-crystals today, and then I'm going to take you through what are the problems of making co-crystals and how mechanochemistry can help in this particular area, right? So, so let's first look at what co-crystals are. Like I said, they are beneficial in drug development. And what are the typical benefits? First, we look at what is the definition, right? This is a lot of controversy about what is a salt and what is a co-crystal. But in the last decade or so, people have settled on this definition. A co-crystal has two or more components in the crystal, right? But these are solids at room temperature, right? All these components are solids typically at room temperature, and they're stabilized by hydrogen bonding. These are not, the interactions are non-covalent and non-ionic in nature, right? So that's what we talk about by being about a co-crystal, right? And so why are we really interested in this co-crystalline system? So there are many advantages, right? First, of course, I talked about solubility. I'll come to that. But they offer a lot of advantages in terms of drug development as well. I took some examples from literature here. For example, you see here, this is a very common drug, carbamazepine, that's been marketed for the last 30, 40 years. And what we see is that carbamazepine, when you actually start to look at the water pickup, how much water it picks up when it's under humidity conditions, it picks up quite a lot of water in 3 to 10 weeks time when it's exposed to moisture, right? About 13 to 14%. So typically during drug development, we do not like drugs that pick up a lot of water, because imagine you have to put it in your cabinet, in your medicine cabinet. It has to have a shelf life of 2 years. We don't want the drug to degrade or interact with water too much. But one of the ways is if you start to co-crystallize it, if you take carbamazepine and form a co-crystal so that both carbamazepine and nicotinamide in this case, or saccharin, if you start to do it, you can see that the water pickup is almost negligible, right? You have altered the physical property of the drug. The active drug is the same, it's carbamazepine, but you have altered the physical property of the drug so that you make it more amenable for drug development. And then the next slide here shows the tensile strength as a function of compaction pressure. What do I mean by it is, all of us take tablets, right? So when you want to do a tablet, you have to compact the tablet, right? You have to put a lot of compaction pressure to get a single tablet out of this. And therefore, you want to see what is the mechanical properties of the drug. Is it amenable to tableting? And what we see here is a simple example of caffeine. And when you start to co-crystallize caffeine, you get far better mechanical properties than you get with caffeine in itself. So that is another advantage of co-crystallization. And finally, the most important advantage, I kept it to the final one, is the solubility advantage, right? So what we show here is, this is one particular compound that I picked up from literature called AMG 517. The concentration of AMG 517 in the blood after AMG 517 is administered to, I believe it's rats in this case, right, as a function of time. This is what we call a PK profile, a pharmacokinetic profile. So we dose the animal and we take blood samples at various time points and see how much has gotten on board after oral dosing of the drug, right? So what we see here, this graph is pretty busy, but I'm just going to draw your attention to these 2 points where, if it just dose the drug by itself, right, this is the pharmacokinetic profile of the drug. It's got very low absorption. But then at the same dose, if you dose the drug as a co-crystal, you get this kind of absorption. So the area under the curve is the total absorption of the drug. You can clearly see that, that is a huge advantage of dosing the drug as a co-crystal, because we can improve the amount of drug that gets onto systemic circulation. So that is the reason why we are very interested in co-crystals, because these have shown to improve dissolution rate and bioavailability, reduce water sorption, improve chemical and physical stability. They have a lot of advantages. But the problem is there are many, many different co-crystal formers, right? How to select the right co-crystal former, and what is and how to take it into development. These are questions that we need to answer, but at the broad level, they offer advantages for what we want to do. So now let's look at, what are the problems associated with co-crystal formation from traditional methods, right? And when I say traditional methods, typically in the industry, for a long time, we have focused on using methods like crystallization, right? It's a great method. You take things into solution and you cool them down or add an anti-solvent and you try to crystallize things out of solution. And crystallization is an impurity rejection process. You get out nice crystals, pure material out of them. Or we try to make it a slurry where you take it into a solvent and slurry it and to get out the final reacted product out. In this case, that's a co-crystal. So what are the different ways of making co-crystals, right? So here, I just want to talk some acronyms, right? Typically, we call the active ingredient, that is the ingredient that actually cures what affects the pharmacology in your body as the active pharmaceutical ingredient or the API. That's the terminology that we use. And then we bring in a co-crystal former. And as I said before, there is no proton transfer here. These are simply the active pharmaceutical ingredients in the co-crystal former are stabilized by hydrogen bonding, right? So there are different techniques that have been used to make these. There are solvent-based techniques, like I said. This is gold standard in the industry, right? Or in many labs too. It's very easy if you think about a wet bench in a lab. You can go take the active pharmaceutical ingredient and the co-crystal former, dissolve it in a solvent, and just crystallize it out, right? Simple enough, right? Or you can do a slurry crystallization where if you don't have enough solubility or you don't have a good solvent where you can solubilize them, you can put them in a slurry and try to crystallize it out of a slurry, right? And then there are solid state techniques. I will come to that in a bit. This is where what we call as mechanochemical techniques, right? In the last 2 decades or so, there's been an extensive application of these mechanochemical techniques to making co-crystals. Mechanochemical techniques, what we can do is we can take, if you remember the definition I told you about co-crystals, both are solids at room temperature. So you can take these two solids in a mill, right? In a ball mill or something like that, and apply mechanical energy to it, right? And therefore you can affect the conversion using mechanical energy, a classic example of mechanochemistry, to make these co-crystals. So you can either do die grinding, or what we can do is what we call liquid assisted grinding. Liquid assisted grinding has also become very popular. Again, this is there's a lot of confusion when I talk to my chemist friends. Liquid assisted grinding is not solvent based, right? It's a catalytic amount of solvent that we use, very, very small amounts. I'll come to that when we are doing the presentation. So then you'll figure out that, the amount of solvent typically needed for chemical reactions is far, far more. We are just using a catalytic amount here to make the co-crystal. So these are the methods that have been used, right? So before going to the solid state techniques and how mechanochemistry is actually being applied, I just want to point out that when we take these and apply our different techniques, right, what happens? So I've taken this classic example from Kairala and co-workers, where they looked at the crystallization of, co-crystallization of trans-cinnamic acid, right, with nicotinamide. These are two common things that we use when we do co-crystallization. So it's a 1-to-1 co-crystal, right? On a molar basis, it's 1-to-1. And they took this in solution at 20 degrees C. So it's interesting. When they do a slurry crystallization in methanol, you get the co-crystal form. When you do it in water, you do not get co-crystal formation. All you get at the end product is the cinnamic acid, right? Then when you do evaporation from methanol, you again get the co-crystal. But when you do evaporation, you don't, cool it down so that it comes out of solution, again, you get only the cinnamic acid. However, when you start to do a dry grinding, you start to get the co-crystal form 1, right? Right away when you put it, you get, that's the only form that you get. Or when you do liquid assisted grinding with methanol or water as assisting liquids, that is not in, it's not in the liquid, but it's a very catalytic small amount of liquid, you still get the co-crystal form 1. So what's really going on here, right? When we start to use our traditional methods, and start to crystallize them, you either get the co-crystal in some cases, or you don't get the co-crystal. But when you're doing solid state chemistry, you reproducibly get the co-crystal form. So the question is, how can we rationalize these results? So, in this paper, they did a really nice work, and this also leads you directly into advantages of using mechanochemistry. But I'll just go through what they did pretty quickly, just to tell you. So you have 3 components here, right? When you're doing a co-crystallization in solution or slurry, you have the 2 components of the co-crystal. You have nicotinamide, you have cinnamic acid, and the solvent that you're trying to do. So this is a three component problem. So what you have to do is you have to develop a ternary phase diagram. For those of you all physical chemists out there, phase diagram is what shows you under what conditions, what kind of end products that you can realize from such a crystallization. So in methanol, you can see that it is a kind of a what we will typically look at is a symmetric phase diagram, right? You have the the molar ratio of 1:1. This is cinnamic acid, this is nicotinamide, and this is methanol. It's a ternary phase diagram, and you have a broad region in which there is the co-crystal and cinnamic acid exist together, or a region in which nicotinamide and the co-crystal exist together. And then when you take this 1:1 system, you see this dotted line. That's where you're going to form the co-crystal. So therefore, every point on this graph represents some kind of thermodynamic ground state. So when you started the 1:1 mole ratio, you're almost guaranteed to get the co-crystal when you do it from methanol, because you are right at that point in the phase diagram that's going to give you this particular product, right? But then when you come to water, you no longer have this symmetric phase diagram. You have a kind of a skewed phase diagram where you're going to get the co-crystal only if you're in this particular region of the phase diagram, right? Because you can see that the 1:1, this is where you start the 1:1 mole composition, but you need you need a slightly different composition of the three components to get to the co-crystal. So, and then when you zoom in on that particular region and take a particular point here, what you really need is what they said is about 68.6 mole% of water, 7.4 mole% of cinnamic acid, 24 mole% of nicotinamide. Or in the weight ratios, it's 9:1:3.2. This is not rational. You're going to just, when you're starting out, you cannot pick this point, right? So then what do we do? Therefore, you need quite a number of experiments when you're going to do it in solution, because you have this third component of solvent. It actually depends upon the relative solubilities of the two components in the solvent system. And then therefore you can have a skewed phase diagram, and you need to figure these things out. And that is why you can get it out of solution, but you have to do a lot more experiments to get it out of solution. However, when you're taking it directly and grinding it, right, you have a cinnamic acid here and nicotinamide here, and you look at the, you measure the melting point of the product that you're getting just using differential scanning colorimetry or something like that. When you start at 1:1 mole ratio, you are almost guaranteed to get the co-crystal all the time, because you simply have this phase diagram where, if you start at the 1:1 ratio, you are going to get there are different forms, polymorphic forms of the co-crystal. Polymorphs are things in which it is identical chemical structure, but they assume different crystalline forms that they pack into, right? There are two different packings here. But this, again, very old work done in cinnamic acid and nicotinamide, but it shows why dry grinding is always going to work, because you are in that part of the phase diagram, you directly go to the product that you of interest that you want. So this, I always like this example because it shows why grinding is such where it works, why it's such an efficient technique, right? I don't need to know anything about this system, in a way, where I just simply put these two components in there and I'm doing, age-old chemistry, right? This is, when human civilization started, we ground things together, right? So we just do that and we get the product. And so this is one of the great advantages of mechanochemistry, if you will. So when you start out with this example, but then of course, there is no free lunch in a way, right? So there are some drawbacks for solid state approaches. Why do we use, we use solvent and solvent-based techniques to make a lot of things, because one is we can make these reactions go forward because they are in the milieu of a solution, they bump into each other, and that facilitates chemical reactions. But a very important point of solids, solution-based crystallizations is also purity, because we can purify whatever is going in, because it rejects impurities as it crystallizes, right? However, the solvent-based methods, we already saw, there are some challenges because there are some kinetic crystallizations, there are solubility differences. And in solid state, we can produce phases that you cannot produce, sometimes that you cannot even produce by solution. So this is all great. These are the advantages of solid state chemistry. But what are the drawbacks? It cannot be used for chemical purification. Garbage in, garbage out. Whatever you start with, that's all you're going to get, right? Magically the impurities are not going to go anywhere, right? And secondly, one of the other things about a decade or so ago, which is not true anymore, which is what I'm going to show you, is that it was really not amenable for scale up for industrial reactions, right? Solution, we know, all the products that we enjoy are all made by solution chemistry. You can make tons and tons of it, and they're not environmentally friendly, many of them, because you use harsh solvents and so forth, but you can scale them up to industrial level production. But in solid state methods, that was very difficult till about a decade or so ago. Now people have started using different techniques. So there are two broad techniques that can help us to overcome this this barrier to scale up, right? First one is you cannot do anything about it. That's, if you want chemical purification, at the very end, we have to put a purification step, right? If you want to do it. But however, the scale up problem can be solved, right? Once you know it's going to work. To give you some idea, a lot of people do mechanochemical reactions in small ball mills in the labs. That's a great starting point, right? We do ball milling, and we can make milligram or even gram quantities using ball mill. But then if you want to go to hundreds of gram quantities, kilogram quantities, ball milling is possible, but it's not the most benign or easiest way to scale up, right? So two techniques have been introduced in the literature for scale up. One is called twin screw extrusion. Another one is acoustic mixing. I'm going to talk about acoustic mixing. Twin screw extrusion too, we developed in our labs probably about 2012, 14 timeframe. We published quite a lot on it. For those of you all, I'm going to talk a little bit about twin screw extrusion because there's a lot of literature about it, and then move to acoustic mixing where there is very little literature about what acoustic mixing can actually do. So twin screw extrusion, this is a very common technique in plastics. We can make billions of pounds of plastics using twin screw extrusion. So that is the great point about it. It's available. It's been used. It's been demonstrated for scale up for many, many years now in the plastics industry. So what does a twin screw extruder look like? It's got a little hopper here where you feed in the material. And then it's got a barrel where you can control the temperature. And then you have different screws, right? The screws can either be co-rotating or counter-rotating. It's like two rotating screws in there. So what this does is the rotation of the screws produces the necessary mechanical shear and stress that you need to drive the mechanochemical reaction forward, right? So you just feed the solid materials. You can also do liquid-assisted extrusion, just like you could do liquid-assisted grinding. Put a very small amount of liquid along with your solid. The liquid can be introduced anywhere in the barrel, and you can start to introduce materials in the hopper, and you have exquisite control, of temperature. There are 3 or 4 zones depending upon the scale of the extruder, and you have a feeding port where you can bring in liquids, and you can have infinite different designs of the screw because each element of the screw thread can be taken out and designed. We have conveying elements and we can have mixing elements depending upon how much shear you want to provide for the chemistry. So you get a lot of control. I mean, you can immediately see it with respect to a ball mill, right? So yes, ball mill you can change the number of balls and in the amount of time that you're milling, but here you can have more exquisite control of the chemical reaction, right? And it's a very short residence time. It only resides in the barrel for minutes. So temperature and other things, you can control a lot of things, a lot of different configurations, and you can get product out. And people have shown it. We have shown it. We can make about a kilogram of material, in less than 20-25 minutes using this kind of technology. And like I said, you have a lot of different extruded designs. You have a high conveying capability if you use these kind of conveying elements with shorter residence times, or you can start to use these, mixing elements. You can see the staggered mixing elements there now here. You can see the mixing elements in these places. And so you convey it, you can mix it more and convey it, and thereby drive your chemical reactions forward. And like I said, this is our lab scale equipment, but you have these huge extruders sitting in industrial locations that you can scale up. So I just wanted to talk a little bit about the twin screw extrusion because it's a known technique. It's been available there. I want to contrast that now with acoustic mixing, which is the rest of the talk. I'm going to tell you about acoustic mixing. So what is a resonant acoustic mixing? So resonant acoustic mixing is a non-contact mixing technology, right? That means that your mixing elements are no longer in contact with your actual product. So this is a big advantage. Before I come to this advantage, I will tell you a little bit about how it actually works. So what happens here is you can have a sample chamber where you drop in, let's say in this case, we want to make a co-crystal. You can drop in your co-crystal, component 1 and your co-crystal component 2 into this mixing chamber. And if you want to add a liquid, you can add a small amount of liquid. And then this whole sample chamber sits atop a resonant, a spring-loaded platform. And the sample chamber and the spring-loaded platform are in resonance. And then what we can do is you can control the time of mixing and the acceleration field that you're actually applying to it. So it's simply oscillating on top of the spring-loaded platform. So this oscillatory force, what it does is that it provides several intense mixing zones, right? For about 50 micrometers in diameter, you can control the temperature, vacuum, and so on and so forth. But this gives uniform and intense mixing zones, right? And it's a low frequency acoustic field. Therefore, even fragile materials can be used, like biologics, if you want to mix, whatever you want to mix. We have done it with peptides and other things. I'll show you some examples later on. So if you bring in normal mixing techniques, many of these fragile materials will degrade because they are very fragile. But here you can do it because you are doing a non-contact mixing and you're doing a low frequency acoustic field to facilitate the mixing, right? So this is a great advantage. So when I say non-contact mixing, why do we like non-contact mixing in the industry? Because your mixing elements are not in contact with it. For example, if you do it in the extruder, your mixing element is in contact with it. Or if you're doing ball milling, balls are in contact with it, which means that there is a potential for some shedding from the mixing element into your product, right? So if you want high quality product, for example, like pharmaceutical industry, where we are regulated by the agency to control our impurities to very tight levels, we don't want any metals or things like that shedding onto that from the process that we use. So if you use a non-contact technique, that's great because your mixing elements are not in contact with what you're actually doing. So this is the big advantage of resonant acoustic mixing, right? So I'm going to show you all how resonant acoustic mixing that we have, this technology, how we have used this to make co-crystals. But just to give you some little bit of history, this was used primarily built for defense applications, right? Where they want to mix, explosives together. They really could not find a technique because you don't want to start doing harsh mixing techniques with explosive elements like TNT and so forth because of the explosion risk that you have during production. So they landed up on resonant acoustic mixing. It was a DoD funded one, and now there's a company called Resonant Mixers, which commercializes this technology basically. So and it's available across different scales, and you can scale it up as well. So given these things, so we said, can we make co-crystals out of that? We were thinking about it in 2014. A bunch of researchers, Armande and co-workers, actually showed in Organic Process Research and Development, this particular paper, that you can indeed make co-crystals of carbamazepine and nicotinamide. You remember one of the examples I showed in the very early slides. It's a nice example system. So they showed that you could make these co-crystals to a 22-gram scale, right? They started in small process development, about 150 milligram scale. And then they showed that these conditions could be employed to make 22 grams, a very nice, beautiful piece of work, which was done there. So then we said that, can we expand on this work, right? So one of the problems I said is, okay, it's great if you know what co-former you're going to use, that's great. And pharmaceutical materials are precious when we start out because each one is synthesized. We can't, 150 milligrams is a luxury for us, right? That's later in development. Earlier on, we get milligrams or sub-milligram quantities from our chemists, right? And we have to do a lot of other studies with it in terms of establishing pharmacology and stuff like that. So we get very small amounts of material. So we said that, can we do it in a high throughput method, right? Can we do, and we don't know what the right co-former also is. So can we do the whole thing on a high throughput plate, right? So what we did is we designed a high throughput plate that will sit on top of this resonant acoustic mixing chamber, right? So we took a 96 well plate, and what we did is you can see that we took a few, caffeine and carbamazepine, theophylline, a few of these drugs. We can vary and put whatever co-formers that we want, like oxalic acid, glutaric acid, malic acid, etc. We can change the stoichiometry. If in literature we have known 2-to-1 co-crystals, whatever it is, you can use whatever stoichiometry you want, and then you can add whatever solvent you want. You can add some methanol, little bit of liquid-assisted grinding if you want. You can run it. So we dispense everything with a robotic platform on a 96 well plate. There's about 6 milligrams of API, or the active pharmaceutical ingredient here, right? What we have done now is, this is because it's a commercial screen. We took commercial materials. We start with 6 milligrams. We can actually do 1 or 2 milligrams as well, because we can run everything on a plate, right? So then what we do is we seal the plate and put it on our acoustic mixing platform, right? And then we do, let's forget about the parameters for now. I'll come to that. There are some parameters that we need to set. And then we, it's gently, oscillating on this. And we can we can control the time and acceleration and so forth. So once it's all done, then we take it out. And then we do an X-ray diffraction on the entire plate, on the high throughput well plate, and see where what kind of crystalline forms that we have, and therefore we can start to judge which of these has given the co-crystal or which are just starting forms that have not produced a co-crystal. Okay, so then when we did this, then what are the results of this high throughput screen? So for this particular screening components that we took, we had nearly about total hits was 81% of the material yielded co-crystals right off the bat, right, in this high throughput screen. So you can see with solvent, we got very good hits, but when we used no solvent, we got some hits, but not great. So this again shows you the power of liquid assisted grinding. I think we all understand now that we need to have some liquid assisted grinding. And again, high throughput is a great way because we don't know which liquid to add, because there is not a clear cut rationale for us that whether we need to add methanol or ethanol or chloroform or whatever have you. So, but in a high throughput format, we don't need to guess. We can run plates after plates if you want to, and it's all done in an afternoon. And therefore we can get to starting conditions where we can form these co-crystals, right? So initially we thought that this reaction may not go forward without beads, milling media. We thought that we might have to use it. And again, you might ask that, oh, once you use beads, it's not contactless anymore. You're actually contacting it. Yeah, initially we wanted to see whether we could do contactless or we needed contact. We didn't know. But as this data shows you, when we had beads or no beads, it didn't really matter. So the acoustic mixing, we don't need any beads, basically. It's beadless milling, if you will. If you just use beads and no solvent, you can get more hits like you show here, but still it's nothing compared to just having the solvent alone, right? So screen had about 80% success rate. We were able to get 13 out of the 16 co-crystals known in literature. We started with commercial examples here. Initially we said the use of beads only for screening. Beads are useful because sometimes they can help with the kinetics of the experiment, right? But we don't need to do it when we scale up. I'll show you what it is. But for screening, we can use beads, and sometimes they do provide some advantages. So what we concluded was RAM provides uniform mixing of the components and hence provides another avenue for co-crystal screening and making. This is actually very powerful because we can, the first time we showed that we can use high throughput techniques for mechanochemistry and how to start to do it. And now you can think of the same thing. A lot of people are publishing chemical reactions. We can easily do the same thing on very small amounts of material and start to see which are the starting conditions that we need to use for scale up, right? And then, so how did this scale up work? First, we took these 6 minutes. So initially we started, if you remember right back here, we took 6 milligrams of the API, and we said, okay, let's do the first scale up. We will do exactly the conditions which provided us with the best, results in the high throughput screen. And we obtained the scale up conversion to the, so we scale it up to 400 milligrams from the screening conditions, right? About a 30-40 fold scale up from where we were from the screen. So what we did is that there are 2 co-crystals of this theophylline citric acid system that we used for the example here. There's an anhydrous system, right, where there is no water in the crystal. And then there's a hydrated system where there is water in the crystal. So what we showed is that we could make both, right? And we said that yes, you can get it with ethanol itself, but if you use beads, it's a little bit better, right? In this case, the conversion goes a little bit faster, but you don't need to use beads, which I'll show you later on. You just need to run the equipment for longer time. Similarly, when you use water as the liquid that in the grinding media, we can get the hydrated co-crystal as well. So these are the co-crystal reference patterns that we took from literature, and then when we started out with theophylline, and this shows that this is the product, basically. And the product now begins to match very well with the reference that is published in literature. So beads do accelerate the kinetics, not needed, but we can accelerate kinetics in some cases, right? And then we can scale up to 400 milligrams, right? And then finally, we said like, okay, that's great. But what are the conditions that we need for scale up? We can't, from an industrial setting, we can't just start doing one parameter at a time. We typically like a design of experiments kind of approach, right? So we'll say that, okay, we want the number of beads, whether we want beads or not, the amount of solvent that needs to be done. In this case, let's say we can, you're using water. And this is where I want to talk a little bit about how much liquid we are really using. So this parameter was introduced by the Frischig group, some time back, where they had a parameter called eta, which is the microliters of liquid per milligram of solid that you have. So suppose I had 100 milligrams of solid, an eta of 0.25 will indicate that there is 25 microliters that we add for every 100 milligrams of solid. So that is the amount that we typically add. It's very small amounts that we add. So if you take in typical chemical reactions, this eta is in the upwards of, typically 50 to 100 in typical chemical reactions, right? That's the amount of solvent that you need, or even more, right? But here we are talking about very small amounts of solvent. So people confuse this as, a solvent, again, as solvent-based chemistries. It's not, right? And many cases, sometimes now we are even seeing that we need eta of 0.04 or 0.05. It's like very, very small amounts that we really need to trigger these reactions to go forward. So then so basically you have a couple of process parameters in the acoustic mixing, how much acceleration that you want to use, how much time you want to mix these things, right? And then you have some other parameters like what solvent you want to add, how much solvent you want to add, if you want to fix the solvent, we want to know how much solvent we want to add. And then milling media like beads, do we want to add them and how much we want to add? So there are a few parameters. So it's better to do a design of experiments kind of work, and we published this. I'm not going to take you into the details of these design of experiments. It's there in our publication. But what we can do is we can quickly arrive at what is the scale-up condition that we need. So finally, we did scale up. And so we did scale up with the beads and without the beads, right? The total mixing time of 6 hours and acceleration of 60 Gs was needed when we used beads, right? At using water as or ethanol as solvent, and we could make 100 grams of the co-crystal, right? And we did this in about 6 hours of mixing time. If you don't use the beads, you got to go longer in mixing time, but you can still get it, right? And then here we show the properties of the scaled up material. Very nicely compares with a reference scaled up material. There's a small amount of unreacted material which is acceptable for what we want to do. This is the thermograms of the material showing that this is theophylline, what we start out with. This is oxalic acid. And then you can clearly see that the co-crystal has a different melting point and it's formed. And similarly, X-ray shows the same thing. And when you start looking at it under the SEM, what you see is these are irregularly shaped plates that you get. Another interesting point that I wanted to do, which is useful for pharmaceutical people, is that you start out, if you look at the blue trace, this is oxalic acid, the particle size of that. This is the size and the normalized volume distribution here. And you can see oxalic acid, the blue trace, has a wide particle size distribution going from 1 micron to 1,000 micron. We just took it out of the bottle that we got. And then you have theophylline here in red. Again, you have a broad distribution, typically, we don't like these broad distributions because you don't want to have very big particles sitting in there. We know it's going to affect dissolution rate. But look what happens when we do the RAM and form the co-crystal. Automatically, you get particle size reduction because you're also mixing and doing all this. So an added advantage, not only producing the material, you're getting intrinsic particle size reduction. Therefore, your solubility and other parameters are going to be slightly better than if you do it with, you have to do a lot of tricks in solution to get to the same point. But here you get it as an automatic side benefit of doing this. So I'm going to run through this example a little bit fast. We recently published this, an example of vitamin C and nicotinamide. We like this system because this system, the one-to-one co-crystal, forms 2 polymorphic varieties. That is, they arrange themselves in 2 different crystal forms. This is called the form 1 co-crystal and the form 2 co-crystal here. And it also forms a 1:3 co-crystal, right? So you can do, so we wanted to see whether RAM could produce the different polymorphic forms and the higher stoichiometric co-crystal. So in other words, can we use RAM to look at variable stoichiometric co-crystals, right? And can we scale them up? It was a different system. So I'm going to quickly take you through it with a couple of points that I want to add here is that, yes, we were able to get form 1 of the co-crystal. In literature, it was already, there was a nice paper which showed that dry grinding, liquid assisted grinding, could form the 2 polymorphic varieties. So when you use ethanol, you get form 1. When you use, this should read as methanol, you get the form 2 co-crystal. Same thing we tried in acoustic mixing. When we use ethanol for about 180 minutes, we clearly get the co-crystal that matches with the literature published X-ray diffraction pattern, right? This is a diffraction, for those of you all not familiar with X-ray, it's intensity as a function of the diffraction angle. And these are published things in literature. If you have, from the Cambridge database, we can take it and simulate the powder patterns and compare it with what we get. Similarly, when we used methanol, we see that we had to do some optimization here. Even if we did it with methanol, we started with 0.5, we didn't get full conversion. We got full conversion only when we went to about an eta of 1.25, right? Or you can do an eta of 0.75, but increase the time from from 180 minutes to 240 minutes, then we could get full conversion. And we can play around with the amount of liquid or the amount of grinding time. And in all these cases, there is no beads, right? So we took these and we said, okay, can we do a scale up to about 20 grams? Exactly same conditions, no problem. We put, and we can scale up form 1 and we can scale up form 2 here, okay? So it's scaled up. And similarly, we could also make the 1:3 co-crystal, and we made it in 50 gram scale. And yeah, we used ethanol. So these were all like within a week or two of starting with it, we were able to show scale up here. And so you might ask, so, okay, we can do it in the lab. What can we do? So there are different scales of this acoustic mixer. We use these scales, in our screening work, in our development work, in our DOE early experiments. We have a RAM 5 that we have just ordered, which can go up to 36 kilograms, and a RAM 55 is commercial. We can go to 400 kilograms of material, right? So technology is amenable for scale up. It can go all the way from screening small amounts to scaling up large amounts. So it's very useful as a mechanochemical tool. So in conclusion, I hope to have shown you all that acoustic mixing is a viable technology for screening up of co-crystals, right? We can even make nanoparticles of this. I've not shown you that. We did a lot of work around that area as well. And co-crystal formation appears to be mediated by highly efficient micromixing, right? This is the key in RAM. So you get constant renewal of surfaces that leads to improved surface contacts and therefore pushes the chemical reaction forward. And acoustic mixing can be considered as, in our opinion, as an efficient, scalable, and environmentally friendly process for the manufacture of co-crystals, an important pharmaceutically relevant material, right? So, what we have done is we have firmly put, what we believe is that we have put acoustic mixing as a clear technique along with twin screw extrusion as now solid state techniques that can be scaled to kilogram levels or multi-kilogram levels if you need. So I showed you this. We can have different techniques. Now we have solid state techniques that can not only do small scale, but also produce large scale amount of material for you. What else can we do with it? Like I said, we do nanoparticle work. We can make nanoparticles. For this, you have to use beads, of course, because you're grinding the particles down. And recently with Frischig group, we have shown that we can run metal organic frameworks. We can make them. We can make them in large quantities. And then we can do metal catalyzed organic reactions. And we have published a couple of papers. We're doing a lot more chemistry with this. And we are doing other pharmaceutically relevant materials like amorphous solid dispersions and so forth. So that's all another day. What we can actually do with this system. There's a lot of applications that we can do. And finally, for those of you all who have taken COVID vaccines, so lipid nanoparticles, yes, we can make lipid nano because it's a highly efficient mixing system. You can take a mRNA and lipid nanoparticle and actually make this whole lipid nanoparticle setup with your resonant acoustic mixing. It's a very topical example, so I just put it out there, right? I just want to finish with acknowledgements. A lot of my co-contributors in our labs, who have helped us, and of course the Frischig group, who Thomas Klav has been our outstanding collaborator for the last 7 to 8 years, and our own management who supports this kind of work, although we are in an industrial setting. So with that, I want to stop here and see if there are any questions. Yes, sorry. I love this. This was not my cartoon. Of course, this is from Professor Lamete in Montpellier, but I love this. It's Yoda with may the mechanical force be with you, right? So we have to change the mindset to thinking about, can we do mechanochemical reactions and run these forward instead of always reverting to solution, because that's the training that we get as chemists or things like that. So I want to stop here and thank Ashley again for the opportunity and see if you all have any questions.
Thank you, Karthik. We have several questions. I'll pick a few for you here. Let's start with, when you make co-crystals by milling, do you ever get an amorphous layer on the surface?
Yes, so I think ball milling, we do get an amorphous layer on the surface sometimes, right? Depending on the material, it all depends on the material properties. Yes, that's possible. I think Nair Rodriguez in Michigan has shown this. In fact, the amorphous phase can act as an intermediate to getting your co-crystal, right? That's been shown. However, in resonant acoustic mixing, since we are using very low frequency acoustic fields, so far we have never ever observed, at least the techniques that we can bring to bear, we have never ever observed a formation of this, amorphous on the surface. So this seems to be purely driven by surface contacts, and that's how the reaction goes forward. In a ball milling, yes, like everybody else, we have also observed that.
Thank you. So I actually received a couple questions about lag not being solvent-based and your comments on that previously. Could you talk a little bit more about the distinction between solvent-based versus non-solvent-based and some of the confusion that arises around that difference?
Oh, okay. So I think that what we have found repeatedly is that, people started with dry grinding in ball mills and things like that. That's how a lot of the early mechanochemistry was done, and that's great. But what they found is that if you add a catalytic amount of liquid, these reactions go forward, right? But the problem is, initially it was said that, we add some solvents into it. So the moment you say that, then people start to think that, then you're just doing solution chemistry, but you're just doing small scale solution chemistry, and it's adding and adding, and you're doing more. So that's why at the very end, I introduced this eta parameter. I know I rushed through some of those things, but the eta is a parameter that, gives you a good flavor for how much solvent is actually being used. It's a microliters of solvent that you use per milligram of solid that is there. Most of our reactions run in the eta at the range of 0.25 to 0.5. What I mean by that is for every 100 milligrams of solid, we put 25 microliters of solution. That's not the solvent level that you use for solution chemistry, right? We are upwards of eta of 50 to 100 for typical solvent reactions, right? We are a lot more solvent. This is a small amount. And what we are finding more and more is that for chemical reactions, especially, is that we can go down to eta levels of something like 0.04 or something. It's just, a tiny drop of solvent that you need. So certainly it's not solvent driven. It's like a catalytic amount of solvent that is needed to run these reactions forward. So that's where we distinguish that these are truly mechanochemical reactions that solvent aids, probably by refreshing the surface or what have you. We don't know the mechanisms yet. There's a lot of speculation, but hopefully we'll iron those things out as we go forward in the next few years. But that's why I want to distinguish. This is not solvent-based chemistry.
Thank you. All right. We also received a couple questions about the effect of the material, the roughness, and the size of balls in a ball mill or the screws in the extruder. Have you any experience with how those factors, which aren't often changed, affect the chemistry that results?
Yes, I think that's quite a deep question, right? Because yes, if you changed the mechanism of how you impart the mechanical force, you can see changes, right? So therefore, if you do something in a ball mill, first let's come to the question of the chemistry itself, right? If you get a co-crystal form 1 or make the co-crystal in a ball mill, there's no guarantee that does immediately you're going to try it in extruder. You have to optimize those conditions for the extruder and so on. And as for the RAM, every time you change how the mechanical force is imparted, you have to optimize it. That's one thing. And the final product, if I understand the question correctly, the final product that you get is definitely going to depend on the properties of that, right? Is the particle size small? Do you have a rougher surface? Yes. In a ball mill, you will certainly get a very different product compared to an extruder, compared to acoustic mixing, although you're forming the same material, right? Even if you optimized it for, like, say, form 1 of vitamin C nicotinamide, I can get broadly different physical properties, right? Particle size, surface roughness, what have you. And they may offer different advantages for you. So earlier on, we didn't have scale-up techniques. Now we have 2. So you got to figure out where do you want to do, what scale-up you want to do. That's, we have to understand those properties, what helps us here.
All right, let's do just one more that your answer there segued into. The question about what scale would you need to match the industrial scales in solution-based drug synthesis with RAM? So how big do you need to go?
Oh, okay. In a way, I kind of showed briefly about the scales right here. So it depends on like everything else. It depends on what you're trying to do, right? So, for example, in a drug synthesis, right? Typically, many of our drugs currently are what we call more targeted drugs, and for targeted populations, we are not looking at more than probably 1 ton a year. Let's put it that way, right? 1,000 kgs a year. For some drugs, which are like, if you're going to do a diabetes drug like Lipitor or whatever, it's multi-ton per year, right? A huge amount that you got to make, right? So RAM, like I said, is you can run it as a continuous process as well. I've not shown it here, but In the 10 kilogram setup, you can run it as a continuous process. So in that case, it's just time. You run it longer and longer and you get it. But if you look at it here, right, this batch size, you can take a 416 kilogram, let's say that's the batch size. That's a total amount of solids and everything that you put in there with a small amount of liquid, whatever you want to do. So you got to run many batches of these, right, depending upon what your volume is. But I don't think we are limited by that. That even in solution, when you use a batch process, you have a certain scale reactor. Okay, you can go 10,000 liters, whatever you want to do, but you're limited by that. And depending upon the amount of material that is required, we keep running more and more. But for purely industrial chemicals, where you want to make lots and lots of it, yeah, that's a discussion. That's, can we match, existing solution capacity, or what can we do? And I think that's where continuous production helps, because we can do it in smaller scales, but you can do it longer and longer. Because you're just continuously making the material. So that's also possible. But really depends on what we mean by scale up, what we're really thinking about.
Wonderful. Well, thank you again for participating in the seminar series.
Thanks, Ashley.
Thank you, Dr. Nagapudi, for that outstanding presentation. And thank you so much for joining us. Just a reminder that this seminar series has included a great group of presenters so far, and we encourage you to check them out on the YouTube channel. Also, we have a great group of excellent speakers upcoming as part of the seminar series, and we hope that you will join us for all of them. Thank you again.